This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWiV, This Week in Virology, episode 461, recorded on September 22nd, 2017. This episode is brought to you by the Defense Threat Reduction Agency, part of the U.S. Department of Defense. The agency's Chemical and Biological Technologies Department hosts the 2017 Chemical and Biological Defense Science and Technology Conference to exchange information on the latest and most dynamic developments for countering chemical and biological weapons of mass destruction. Find out more at cbdstconference.com. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Hello, Vincent.
Lovely day, isn't it?
It's a fantastic day out there.
So it's all sunny, no clouds, right?
Oh, I wouldn't say that. If you just turned around,
I don't
you
want
could see for yourself.
Well, if I did, I would be detached from the mic.
It's partly cloudy, but the humidity is low. The temperature is perfect. The wind is down. Looks great.
It's currently, I have a new weather app, 83. Oh my gosh, it's in Fahrenheit. I'm not going to use this one. Nope, no way. We're going to go back to the old one until I can figure out how to switch. 26.
I was going to say 27.
But we have, the next week is good.
Yes.
Good weather.
Yes.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's 89 degrees Fahrenheit here, which is 32 degrees Celsius. When I first saw the 32 degrees pop up, I thought, no, it's not cold. But
Nice.
32 degrees Celsius. Yeah, and we also are going to have nice weather the rest of the week.
It's so warm. That's beautiful.
Yeah.
Wow. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
Let me take a guess. 92?
No,
it would be less.
88.
4 degrees. Said last.
Hey, we got you beat.
31 Celsius and partly cloudy. And I must point out, actually, our listeners are going to get confused because this is going to get posted late, right?
Yep, a week late.
But relative to the reporting, it is the equinox, right?
It is.
Today.
At 4 something. It's not yet. It actually is at 4 something.
On the 22nd, is that right?
Yeah, that's right.
So yesterday was the first day of fall.
Official. Yeah, but today is the actual day. Today is the day the Druids celebrate.
I know this because of the banner on the Google search engine.
Exactly.
It's where my news comes from.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, everyone.
Hey, Brianne.
It sounds like Madison is the coldest because it's only 81 degrees Fahrenheit here.
Not by much. Not by
Or
much.
27 Celsius, but it's also going to be fabulous here over the next week.
So we had our microbiology and immunology retreat right near you, Brianne. There's a hotel, I think it's the Wyndham or something, right next to your campus, I think, right?
Yes, it is right next to our campus.
Yep.
I stayed there when I was moving.
Yep, we had our retreat there. Everything good with your course, Brianne?
Everything's going very well with my course. The first virology exam is on Monday.
Wow.
They're studying lots now.
Yeah, how many ruined lives this weekend?
That would be 22 ruined weekends.
22.
Meet the Microbiologist is a podcast that showcases the people behind the scientific discoveries. Each guest discusses their research in one of the cutting-edge areas of the microbial sciences: genomics, antibiotic resistance, synthetic biology, emerging infectious diseases, microbial ecology, public health, probiotics, and much more. Meet the Microbiologist is hosted by Julie Wolfe and was previously called Meet the Scientist, which was hosted by Mary Buckley and then Carl Zimmer. Listen to Meet the Microbiologists at asm.org/MTM. Alan, who's not here, reminded me that it was 9 years ago that we recorded TWiV 1.
To this day.
I'm not sure, was it a Friday?
That's what he said.
And he said it dropped on Monday. Now, the dating is going to be confusing because this is going to be released a week late.
Yeah.
But Sunday, September 24th.
I can't get it out of my head how impressed I was over the fact that you asked me to do this.
Kidding.
No, because I'm not a virologist. You knew that. And I'm still not a
virologist. You're a communicator.
You know, this is true, but I was still, I guess, in awe of the fact that that you could actually communicate in this way and still.
I think it took you 8 years to find that out.
It's still sinking in, to be honest.
So wait a minute. On this event, maybe you could recount the story to me, because I recall something about Vincent having the idea, walking in from the parking lot with you, and you said, "Let's do it," and you did it that day, something like that.
No, that sounds great, but I had thought of the idea the day before, listening to This Week in Tech, and I said it would be cool to have a This Week in Virology, and I thought of Dickson, good teacher, great teacher, and so the next day I ran into him coming in from the garage.
That's true, and I didn't get hurt too much.
I said, Dickson, I have an idea. And he said, good, let's have lunch. And that's Dickson for you.
That's true. It's true.
So I didn't get a chance to tell him in the morning, but I went to lunch. I said, I'm thinking of doing a podcast, a recorded show where we have conversations about virology. And he said, let's call it This Week in Virology.
Did I actually say that?
The same title. I said, will you do it? He said, yes. So I don't know, a week later or a few days later,
And tell him the other part of that story then.
Well, we went to your office with the nice view of the Hudson.
Not bad.
And I had a recorder and 2 mics, and I didn't plug it in properly, so the battery ran out. The card filled up because it was too small. So we only got halfway through and had to come back the next week, and we had to finish the recording. And by then we'd forgotten what we said. So if you listen to it, there's a lot of duplication. But
that's right.
And then while I was editing it, I guess it was on a Saturday, I thought of this, the thing at the end, that another TWiV is viral, and the other thing, the podcast about viruses, the kind that make you sick.
Yeah.
And I ran it past my daughter, who was at the time, 12 or 11 years old. And she said, "That's great, Daddy." And then,
can I have some more
mousse? I don't even know if she listened to me. So I went with it. I thought it was a little clever, and so we still use that.
That's good.
9 years, so
9.
That's pretty cool. I have to say, everyone, so next year will be 500th episode, and in September it'll be 10 years. And someone invited me to Berlin the same week as TWiV 500. I said no. I said no. I have to go to TWiV 500. No, it's already set in stone.
It's in Austin.
It's already set in stone.
It is in Austin.
And so we're going to do that. That'll be fun.
Yeah.
Okay, we have some follow-up. First one is from Xander. Hello, TWiV team. I enjoyed the latest episode 459, especially the polio part. I've been working with this vaccine and developing new ones. As with aircraft crashes, there is not a single thing going wrong, but a series of small things end up in allowing Murphy to wreak havoc. Some links to the companies with recent problems in keeping containment. So he or she, I don't know what Xander would write. What do you think, Kathy, would be? Do you have any?
I'm not sure what you're asking. Wait.
Gender.
I'm saying male. Okay. Quite often that's basically a nickname for Alexander.
Oh.
Yeah, okay. I'm just preempting you telling me I should not think it's a guy. That's all.
Sorry.
It's all right. So possibly Xander, a male, sends a series of links. First, from the paper you covered, he sends a link. And these are mostly in Dutch, but some of them have English. And then he or she has a link to another earlier incident. And continuing, the WHO limits the amount of manufacturers of wild-type polio. Currently, there are 4 worldwide with a license to produce wild-type-based vaccine, and no new ones will be issued. We have GlaxoSmithKline, Sanofi, SII, Bilthoven Biologicals, AJ Vaccines, AS, former Statins, Serum Institute. As you stated, a lot of research is being performed in new vaccines, such as a Sabin-based vaccine, as mentioned as well, and a progress report, a couple of links there. Also, new polio strains made by the CDC and NIBSC, that's Andrew McAdam. NIBSC, National Institute for Biological Standards and Controls, like the FDA of the UK, are being tested as alternative genetically weakened. All in all, the capacity of polio vaccine production currently is too low to keep up with demand. New vaccines are needed, but we cannot wait on them. The need for more stringent containment is clear, but left by the WHO to the local governments to implement on their timetable. To make matters worse, new vaccine concepts based on GGO are now already made a BSL-3 agent, while the wild type still remains BSL-2. So near eradication, we need to keep up the work and accept some risk. With kind regards. What's GGO?
I'm thinking that's some abbreviation for genetically modified organisms.
Yeah.
Okay. So this is a European thing, right? That if you're messing with a genetically modified, or I'm making this up, okay? But this is the best I could make out of that. That if it's a genetically modified organism, it's automatically BSL-3 or something like that. So, but I'm not sure about that. This earlier incident that he points out, whoa. Somebody mistakenly dumped 45 liters of concentrated live polio into the environment, I guess, in the waste.
Yeah, I think we covered that. That was in 2014.
Did we cover that?
Yeah, I'm pretty sure. Anything of that magnitude, yeah.
Man.
It was accidentally, the wrong valve was opened,
Yeah. But, the interesting thing is that they couldn't find anything downstream. It dilutes out really fast.
Yeah, it sure does. The solution... Well, the story, right?
Turned out not to be true, by the way.
Rich, can you take the next one, please?
Sure. Steen writes, to my favorite pedants. This is coming from another pedant,
by the way,
I think.
Did he spell that right?
The spell checker is saying it's wrong here. Isn't it pedants?
No, the spell checker wants to put in pendants.
Oh, pendants.
Spell checker's just ignorant.
My favorite
How can it be that doesn't know pedant, right?
Yeah, well, I was just being pedantic and suggesting that we check the spelling.
All right. Are we finished now?
It's in the Apple Dictionary. It's correct. Pedant. All right. Sorry.
How appropriate. This is great. I enjoyed the discussion. I enjoyed the discussion on TWiV 459, especially circa 47 minutes. Props to Vincent for referring to N. benth, Nicotiana benthaminiana. I'm going to mess this up. Benthaminiana.
No, there's no N in there. Benthamiana.
Benthamiana, thank you. As tobacco-like rather than calling it tobacco, which is Nicotiana tabacum. I appreciate his careful use of terminology, including avoiding reference to expressing proteins in plants. The polio paper you discussed on TWiV 403 used N. tabacum proper plus lettuce.
By the way, we didn't mention that episode.
No.
Yeah, TWiV 403 from last August, 2016. It's not easy being vaccine.
N. benth was originally adopted for research use because of its nice broad leaves and because of the broad virus susceptibility of a particular strain. This susceptibility is associated with a loss of function of an RNA-dependent RNA polymerase involved in silencing amplification. The mutation appears to confer advantages in desert habitats. See the recent study by Bally et al. It gives a link. The paper is not open access, unfortunately, but it is accessible on another site. See also the accompanying news and views piece. That's actually very interesting.
Let me say the title of that. The paper is "The Extremophile Nicotiana benthamiana has traded viral defense for early vigor." VIGOUR. Love it.
So I have color war.
Go ahead.
So the bottom line here is pretty cool. We show the lab strain of NB is an extremophile originating from a population that has retained a mutation in the polymerase gene for 0.8 million years and thereby traded its defense capacity for early vigor and survival in the extreme habitat of Central Australia. That's the conclusion. Cool. All right. Sorry. Go ahead.
All right. I have never heard of transmission of agrobacterium by an arthropod vector, one of the things that we discussed the possibility of. It is a soil bacterium. Many strains lack the tumor-inducing plasmid carrying the opine synthesis genes that plant cells are hijacked to express for the metabolic benefit of the microbe. Unfortunately, the floral dip stable transformation method that Kathy mentioned does not work for most plants, probably because their female germline is less accessible than it is in Arabidopsis thaliana flowers. I believe that Caliber Biotherapeutics, which Dickson visited and also linked to for his pick of the week, has been renamed to iBio Inc., and he gives a link to it. Note the potential for confusion with iBiology.org, the makers of those wonderful science videos. Agro-mediated transformation is not the solution to all problems. Viral vectors are still important, as is evident in this news story from a few months ago. And I just want to, he gives a link to that, and that's a pretty cool story.
Isn't that out of Gainesville, right?
Yes, the guy, Dawson, Ralph Dawson, I think it's Ralph, who is one of the big sort of plant virology. Bill Dawson who is the plant virology guy in Florida. There's this, is it citrus greening disease?
Yes. Correct.
That's trashing the Florida citrus crop. That's a bacterial disease. And they're trying to breed resistant plants and that kind of stuff. But Dawson's engineered a virus. I forget what it's doing.
They identified a defensin from spinach.
That's right. Yeah,
that kills the bacteria.
Right. Cloned a defensin from spinach into this virus. Yeah. And the way they get it into the trees is that they have trees that they graft infected bits onto the orange trees to disseminate the virus.
It's citrus tristeza virus, right?
Yeah, which is a positive strand RNA virus filamentous.
Yeah, so we have a saying, which I'm sure we've expressed before on this show and on others too, that successful systems attract parasites. The moment you monoculture anything, that's considered to the invader as a successful system. So that's how these problems get out of hand very fast.
Sure.
The interesting thing here in this article, Rich, and everyone, OJ made from transgenic fruit will carry a GMO label, but juice made from a vector-treated tree would not. So that's interesting in light of our recent discussion.
Very interesting. Yeah. Well, that's because of Luther Burbank. Way back in the day, he was the one that grafted all these things together to make crazy hybrid fruits. And so that became a normal thing to do. And so that's not considered an experiment, basically. That's,
Lastly, Steen says damage from Irma, and he's got that linked, recently added to the economic losses from the citrus greening crisis described. And that's an interesting link because Irma blew through and before the crop was ripe and blew 75% of the oranges off the trees. So they're having it was going to be a good year. Now it's not.
No.
Best wishes, Steen.
It's a very informative email.
Yeah, this is great.
Yeah.
Loaded with stuff.
Kathy, can you take the next one?
Yes. Hamish writes, "Hi, Vincent, Dickson, Alan, Rich, and Kathy. Greetings from a semi-clouded Amsterdam, 17 Celsius. Go metric, U.S.
Trying.
With a few spots of blue sky here and there, a light breeze, an enjoyable autumn late afternoon. I'd like to mention that in the Dutch news coverage, the operator was identified as being male. The company is called Bilthoven Biologicals, which mentions the incident on its website. And he gives a link to the English version, which is at the bottom of the page. And he has a couple other links about it, which I didn't have a chance to check up on. But he says Google Translate does an okay job on these. So evidently they're all
pretty much
Dutch.
They're pretty much redundant of everything that we've already discussed. And in fact, the translations are pretty amusing in places.
Okay. P.S. 1. While I'm not an academic, I still enjoy listening to TWiV a lot. Thanks for making virology and science in general a bit more accessible to me. P.S. 2. Vincent. The Dutch stad sounds more like Stadt, though. Your version has a more German feel to it. Anyway, not half bad considering the way you butcher the names of the authors. Smiley face. The UI sound is difficult. Another smiley face.
Right.
PS3, Dickson, ? The Netherlands is small, but Bilthoven and Amsterdam are still 50 kilometers apart. With 20,000 inhabitants, it's not considered a hotspot for transit traffic, for example, tourism. And that's from Hamish.
Thank you, Hamish.
Yeah, not our Hamish, but another one.
No.
Now, look, at least I tried to pronounce.
Yeah.
I have zero training in Dutch.
This is true.
Butcher. I also liked what Hamish says, making science in general a bit more accessible.
Just a bit more. That's right.
Not a lot. Just
That's right.
I guess we have to be a little clearer. Right?
Yeah.
All right. Well, speaking of clearer, our snippet is an interesting paper in eLife called "The Readability of Scientific Texts is Decreasing Over Time." It's from a group at the Karolinska. Now, the one thing I do not like about eLife papers is they put the author affiliations at the end of the article. And here on TWiV, we can't be scrolling all the way up and down. We can't do that. We see the names here, which are Plavin, Sigray, Matheson, Schiffler, and Thompson. So
But if you have a paper copy, you might hear mine rustling.
That's right. I hear yours rustling. That's right. It shows you why a paper copy is good, because you could get to the end much faster than I could.
I did.
Scrolling, yeah. The idea here is that there's some evidence that, this is wonderful. There have been trends towards simpler language in U.S. presidential speeches, novels, and news articles.
Yes. I have that highlighted.
And for this year, it's even simpler.
Yeah.
But there's some studies which suggest that there's an increase in science writing complexity. So they wanted to rigorously look at this. So what they did is they picked 12 fields of research, and these include some of the ones we love, like microbiology, immunology, neuroscience, plant science, etc. 12. They picked just 12, and then they identified 123 journals, including Nature Science, New England Journal, Lancet, PNAS, JAMA, and they downloaded from 1881 to 2015.
1881. I love that.
It's old. There weren't many back then.
I love that.
709,577 abstracts.
Fantastic.
I wonder how you do that.
Well,
you have to have some kind of script, right? Or maybe there's already something.
There's an app for that, probably.
And then they use two different methods for assessing the readability of these abstracts. One is called the Flesch Reading Ease, FRE, which basically calculates the number of syllables per word and the number of words per sentence, with the idea that more syllables, it's less readable. And then we have the new Dale-Chall readability formula, where it uses the number of words in each sentence and the percentage of difficult words. And this is difficult is defined by a list of words that are
interesting.
And Brianne, that reminds me of that upgoer thing, right?
It does. It reminds me of it too. We talk about papers every Friday in my virology course, and the students have been complaining about the difficulty they've had with reading some of them. So I told them about this paper this morning, and they were very excited. They think that it must be very true.
Yep.
So basically, these FREs and NDCs, you can use a computer to figure that out. We could take any of our papers. In fact, they do it for this paper at the end of the article. They calculate the FRE. And there's clearly a decreasing trend of readability based on these two metrics from 1880 to the present. It's a graph. This is open access, so you can see it. It goes from a readability, a FRE of 40 in the 1880s to 10 currently. And you may wonder what that means. So we'll have to scroll all the way to the end to tell you, because that's where it is.
I have it written right here.
Tell us.
100 is something that a 10 or 11-year-old can read. And 0 to 30 is something that college graduates can read. So back in 1880, things were around 40. Now they're at 10. And 0 to 30 is college graduates. And then they said somewhere, it's not shown on this graph, that there's a lot of papers that are negative readability on this score. So they're not even readable by college grads by that criterion.
Yeah, it's 22%, or couldn't be, or beyond college graduate level English. You know, I looked at the recent abstracts that I've written, and I have to agree that it's really hard to read.
I mean, they didn't have page charges back then, right? So they didn't have to worry about saving words to abbreviate things or to, put it into a context that only the people reading it would understand.
These metrics take into account the number of words, right? That's part of it.
Number of words, but when you abbreviate to save on a freight train sentence, so to speak, then that's what's happened over time, because we can now abbreviate a lot of things that we couldn't before, because now we know what they are.
Yeah. Well, I think there are many possible reasons or explanations for this trend, and they look at two of them. One is that there are more authors per paper now than there used to be.
That's right.
And the other is that we're using more jargon.
And more jargon, and that's the abbreviated stuff. That's exactly right.
They conclude that there's no correlation between the number of authors and and the readability, but the jargon is an issue.
Jargon is horrible.
To me, I mean, this is all cool, but to me, these quantifiers don't really capture what makes papers unreadable for me. Okay. It's more, and we've complained about this a lot on the show. It's this having to cram a lot of stuff into a small place.
Yeah, that's
leaves a lot of stuff unsaid.
That's the deal.
And rather than a nice description of what's going on, you have to interpolate a lot using your knowledge. And actually, I wind up, doing a lot of research on the internet to understand what this and that means, to understand the paper.
For sure.
And relative to the 1880s, we've talked about this before. The older papers have a much more sort of conversational
for sure.
Style to them. I said, we had a cup of coffee and then we talked about it for a while. And it's just, a much, much easier read.
Oh, absolutely. If you look in the 70s, at any, like a virology paper, I'm going to pick one out here. Let's see. Because I was just looking at some today. Here's a 1963. Let's see if they have the abstract. No, there's no abstract. Oh, wait, PMC free. Here we go. Yeah, some of them were very short. Here's an abstract of a PNAS paper from 1963. The widely accepted thesis that RNA synthesis is required for phenotypic expression of DNA-borne genetic information. I think that's a pretty hard sentence.
Yeah, that's pretty hard stuff.
So, it doesn't.
Yeah, Brianne. I think that this is an underestimate, given it doesn't really get to density like Rich was mentioning, but it also doesn't really get to how much a word might have a specialized meaning. So maybe it's a common word, but has a sort of a really special meaning. In our other paper that we're going to talk about, there were a couple words that I had to think carefully about that might be common, but they're used in a really specific way.
Here.
Yeah. One of the entertaining things here is in this figure 6, they pick out a number of words, right, that they associate with increasing difficulty and look at their usage over the years, and they're really interesting. Okay, robust.
Yep.
Right, is just skyrocketed over the last few years.
For sure.
That's right.
For sure.
Novel.
Exactly.
Significant too.
That's right. That's right.
So,
that's right.
But you were talking about readability, and you know that I'm often drawn to figures that are really easily understood versus those that are not. And this figure, it took me a long time to look at. I was annoyed that they didn't use the same y-axis for all of them. They didn't seem to arrange them in any kind of order. Like appears has been decreasing, but it's just kind of mixed in with all the others. They're not in alphabetical order. There's no rhyme or reason to it. So
it's true. Wonder why appears is decreasing.
Yeah, I wondered the same thing. The only thing I could think of is that there's this pressure to be definitive,
Yeah.
So you wouldn't say that something appears. You'd say, this shows.
Yeah.
This is.
Yeah. But then suggesting is on the increase.
Exactly.
So I would have liked them all to be on the same scale. I think this is misleading.
Right.
I think that there are probably other reasons, too, for less readability, right? I don't think it's just this. I mean, this may be a main component, but I think there are other things. So, for example, I tweeted out this article, and Paul Benash said, how about increase in sophistication of the science and the volume of work in a paper probably contributes to decrease in readability. That's certainly part of it too, right?
True.
Yeah, and I liked how in Figure 3 they were kind of showing differences between fields, which might get to that same thing. You know, that some fields have been declining in their readability much faster than others. It seems like molecular biology, genetics, and biochemistry has really lost readability.
Yes.
Right.
So now this is not good because it makes science less accessible, right? I mean,
It's true.
to non-scientists, but even, across fields. Today we're going to talk about a neuroscience paper, which I found less accessible than papers in my field because a lot of the words that were used.
That's right.
So what can we do?
Duh.
They have a few suggestions which are worth going over. One is to have lay summaries in journal articles, and some journals are starting to do that. We had to write one for PNAS recently. And then this is funny. Some have noted that scientists are increasing their communication with the general public through social media.
What?
Some have noted. I don't know what world they're living in. And this trend could be encouraged. Really. It sounds like they're not sure.
Those phrases are both passive voice.
Yeah.
If they had made them active
Interesting.
voice, they would have
Interesting.
been more accessible.
Yeah.
So
then they propose
Go ahead.
to make scientific communication a necessary part of undergrad and graduate education. And it's really interesting because I was at Penn on Tuesday and I spoke with a small group of grad students and postdocs about communicating science. And this is one of the things they said, why can't we have courses in science communication?
Right.
Good idea. Do you have any at Drew, Brianne?
We do not have specific courses in science communication at Drew, but I have been trained in teaching writing for my first year seminar, and almost all of my courses have writing intensive designations. So we're trying to incorporate it into our science courses. But I agree, we need to have specific courses.
Kathy,
Yes.
do you have any at Michigan?
Yes, we do. And it's taught primarily by somebody in pharmacology to all of the second-year graduate students that want to take it in the biomedical sciences. And then it has breakout groups partway through the semester that specifically work on a grant proposal. So I've done the breakout groups for years.
Is that Pavel?
The person
Yeah.
teaching it? No, it's Lori Isom.
Okay. I thought it was the fellow. Sorry. Go ahead.
UF had probably still has a grant writing course that was very popular for incoming graduate students. And I used to teach I used to give a couple of talks on giving a talk.
I'd love to teach a course on communication of science, but using podcasts and videos and blogging and social media, right? That would be cool.
That would be great.
Do hands-on stuff.
Sure.
Hey, you could do the upgoer thing that we talked about. You could do your elevator talk, practice those. It really could be a fun course, right?
That would be a lot of fun.
And you could get, you could do, there's a lot of cool things you could do with that.
I wanted to just add to this conversation. It's just not just about science per se, but I've been forced by my new subject area, namely urban agriculture, to get involved with architects. And architects have a language that they use English words. None of them mean what you think they mean.
Okay, you have an example?
Yeah, tectonic.
Those are the plates of the earth, right?
Exactly.
So what do they mean?
However, they probably, well, I can just say they probably mean where the building sits. Okay. They talk about architectural tectonics in a way that's totally unintelligible unless you've had the course, and then they define it for you. And then after that, everybody accepts the meaning, but it's just exclusionary in terms of their language choices.
Well, tectonic does apply to building or construction, according to the dictionary.
Appreciate that. But yeah, because they wrote that into the dictionary.
You think?
Well, I mean, I listened to an architect once give a treatise on not the history of architecture, but rather the meaning of architecture for the living environment. And it was eloquent to the nth degree in the choice of really $10 words, I would say, and maybe some $20 words thrown in. And it was almost, I needed an interpreter to sit down with me and say, and by that he really meant, well, I was astounded at what I didn't know. And I thought I understood, the English language as well as anybody else. But their choice of words and the way they describe things are just, I would call it not purposefully obtuse. It's purposefully exclusionary so that they can have a conversation without being interrupted maybe or something. I never could figure out why that was the case, but
sounds like immunology.
A little bit, or the choice of words to name genes.
Would be interesting to compare different fields in their readability.
I would love to see them tackle the architectural literature and just to see what they come up with, because even though it's English, it's just not your English, it's theirs.
They suggest making an R index for writers, like the H index for publication citation. That would be a measure of your readability.
Oh, I got it.
I think that's a good idea. I would love to see that done.
I mean,
I like that.
It would certainly improve your chances of getting funded through the National Institute of Health if your grant was more readable.
Well, if someone had a website where you could paste in text and get a readability index, that would be very useful.
It would be fabulous. Or hit the word count and then hit the readability count.
Probably it's an easy formula, right?
Yeah, that must be doable.
True.
In fact, it's
in the paper.
A metric.
Said that somewhere is saying that you could, we can all do that. And I thought, well, I haven't seen how to do it in Word. And then I thought, well, if Alan were on, he would tell us he doesn't use Word and he can do it in his way.
Right.
So, for example, the Flesch RE is very simple. The number of words divided by the number of sentences plus the number of syllables divided by the number of words. That's it. That is pretty simple. So you could easily do that on a web page.
So it looks like there is a web page that I found that will do it.
Cool.
I can paste in some text, or I can subscribe and do all the text I want.
I'll be darned.
Interesting. Can you put that link in?
I'm putting it in right now.
So this paper, the article that we just discussed, has a free score of 49, and the abstract has a free score of 40. Which would, in terms of their graph, 40 would be like in the 1800s. When it was good, when it was readable. So they're saying that the article is readable.
Have you ever read Origin of Species?
No, not all of it, parts of it.
Exactly.
It's
archaic.
It's a pretty good read.
It's a pretty good read, but it's extremely difficult.
Have you read Ulysses?
That's not in English. That's just using English words.
It's tough, isn't it?
Yeah, it's very tough.
Well, that's cool. We got, and now I suspect that speaking is a different thing altogether, right? And you can't just transcribe like a TWiV episode and see how readable it is because it's people talking.
Yeah.
Do you think that we use a lot of jargon?
No, we don't. We try not to do that because
I'm
your jargon expert.
We're not all that successful.
Listen, that's why you have me on this show, isn't it? Because I'm not familiar with all that stuff. So I ask you, what does that mean? What does this mean? And I think sometimes everybody does that because that's a term that you're not familiar with. So we try to limit it. And then you describe it. And once you've described it and defined it, it doesn't become jargon at that point. It becomes part of the conversation. So,
but people who listen continuously can figure it out. But people have written that we do use a lot of jargon.
Right. But notice how fast the word desoxyribonucleic acid became jargon and then everyday language.
Not just DNA, right?
Exactly.
But I think maybe not everyone even knew what DNA stood for.
They do now.
And now people use it all the time in a way that drives me crazy.
Exactly. The DNA of the organism or the DNA of the company.
Do you mean like
no,
the DNA
it's in
of the
our
company?
DNA.
Yeah,
it's in our DNA to talk about
Well, they
jargon.
Use the word ecology the same way, and I reel it up.
This company's DNA is based on. Right?
Yeah.
You don't like that? I don't like it.
The ecology of that organism.
So I sent this to Jane Flint yesterday, and she loved it, and she said, I just went to a seminar where the jargon drove me crazy.
Yeah, right. Sure.
So I can see Jane going to seminars and just being crazy at each one because of the jargon, because she's particular.
So I just pasted the abstract from my most recent publication into this little machine here, and I got the lowest grade available.
Oh, no.
I'll try another one.
That's right. Try again later.
All right, while you're doing that, let me tell you about the sponsor of this episode, the Defense Threat Reduction Agency. Imagine an everyday, inexpensive drone you could buy online modified by terrorists to spread chemical or biological weapons over a crowded football stadium or holiday parade. plague, VX, sarin, weaponized influenza. How could we prevent a scenario like this from happening? How do we counter the effects? Where would we treat the victims? Join us in Long Beach, California, November 28th through 30th for the 2017 Chemical and Biological Defense Science and Technology Conference to exchange information on the latest and most dynamic developments for countering chemical and biological weapons of mass destruction. Collaborate with more than 1,500 scientists, subject matter experts, military service members, industry partners, and academic leaders from across the globe who are committed to making the world safer by confronting chem biodefense challenges. Part of the U.S. Department of Defense and charged with safeguarding our warfighters and our nation, the Defense Threat Reduction Agency's Chemical and Biological Technologies Department hosts this important conference and brings together the best and brightest from around the world. Please join us to share your important ideas. For more information and to register, you can go to cbdstconference.com. That's all one word. And you can also stay up to date on Twitter and Facebook. On Facebook, you can just search for DODDTRA. And on Twitter, it's CBDST Conference. The 2017 CBDST Conference, today's innovation, tomorrow's warfighter capabilities. All right, we have a paper for you.
Wait, you don't want our scores on our own
abstract?
Yeah,
you got it. You all did it. Go for it.
Well, I had to take a screenshot because I wanted to also test the lay summary. So I got a 26 on the flesh reading ease on my most recent one. And then I had to reboot and paste it again. I have to click on measure readability. And it has a 34. The lay summary has a 34 score. So it's...
What was your letter grade?
E for the first one and D for the second one.
Yeah.
Yeah. I used too many long words.
I think
encephalitis, neuroinflammation, enzymatic.
I think if you pasted in today's paper's abstract, you would get a pretty low score too.
Yeah.
So too.
Okay.
And Rich, you got the lowest grade possible, right?
Twice.
Okay.
I think
nice. I'm going to actually find a chatty email here and see if I do any better in regular conversation.
All right. Our paper is published in Cell. It is entitled Lifelong Genetic and Functional Access to Neural Circuits Using Self-Inactivating Rabies Virus. Comes from the MRC Laboratory of Molecular Biology in Cambridge, UK, and the authors are Ciabatti, Gonzalez, Rueda, Mariotti, Morghese, and Tripodi. And this is about a vector used to study the nervous system. And I got interested in this because I'm on the Institutional Biosafety Committee of the medical center, and I was at a meeting last week. And every meeting they have something called, educating the members of the committee. And it had one person who runs the viral vector facility at Columbia, which is located on the new campus at Manhattanville. And she talked about the different vectors they can produce. And she went through all the vectors, a historical description of the vectors used to study neural circuits. And this is the most recent one. And I was just amazed. And so I said, we have to do this because it's so cool. And so we're going to tell you about this. There's a lot of background we have to give you. First of all, it is, so it's based on rabies virus, a negative stranded RNA virus with an envelope. Of course, it's bullet shaped, causes rabies, no doubt. And it's been modified in a way to make it a good tracer, as you'll see. And that's done because we have infectious DNA copies of these genomes, and we can add in and take out genes at will. Now, the rabies genome doesn't encode all that many proteins. In particular, there's a RNA polymerase, there's a glycoprotein, and there are other accessory proteins as well. What is it, 6 proteins? I'm thinking off the top of my head.
So matrix, N, P,
L,
polymerase,
glycoprotein.
That's 5.
5 major proteins.
That's about it.
Okay, so you can modify this virus. We know it's got exquisite neural specificity, And so many years ago, people decided to use it as a tracer. Now, why do we want to do this at all? There are 100 billion neurons in your brain, Dickson. And each neuron can have, on the average, 7,000 synaptic connections with other neurons.
It's mind-blowing.
1 quadrillion total synapses.
That's incredible.
And of course, if you want to understand something that's wired, you got to figure out the wiring.
You do.
Basically. And that's what people have been trying to do.
Yes.
And for years, they have used various chemicals to trace neuronal connections. These are called transsynaptic tracers.
Right.
You know, you can put these chemicals into one neuron, it will cross the synapse and go to another, but they're not great. And there's one of the papers that we'll put in from 2007. They discuss why these tracers weren't working. This was one of the first groups to make a rabies virus to do what's called monosynaptic transmission. So you can put the virus into one nerve cell and it will cross the synapse to a second, and that's it.
That's the 2007 paper that you're talking about.
Yeah, this
was the first to do that. Right.
Yeah, this is called monosynaptic restriction of transsynaptic tracing from single genetically targeted neurons. Okay, and they say basically you can't do this with anything except a virus. A virus is a great way to do this, and
right.
Rabies. You may have heard Lynn Enquist over the years speaking about using herpes, not alpha herpes viruses. You know, swine. What is it?
Porcine.
The pig virus that he uses.
Cholera?
No.
Pseudorabies.
Pseudorabies virus to track connections. But apparently this has advantages over, because they say in this paper, rabies virus has significantly lower cytopathicity and greater infection efficiency than the other widely used family of tracing viruses, the alpha herpes viruses. And that's what Lynn Enquist and others have used. It crosses synapses exclusively in the retrograde direction, which means towards the cell body, right? And it's specific to connected neurons, not to adjoining neurons that are not connected. So these are why rabies viruses are good. Now, what they did in that paper is they deleted the glycoprotein of the virus. Okay. Now, when you do that, of course, you can easily make particles by putting the altered genome into cells. But, of course, they can't infect anything because the glycoprotein is needed for attachment to cells. So what they then did, what they do is they supply another glycoprotein and its receptor along with the virus when they put it into neurons. So they have this G protein deleted rabies virus. They micro-inject that into a neuron. So they stereotactically inject the virus into the brain of an animal, say, or you could do it in cell culture as well.
So what do you mean by stereotactically inject?
So you hold the mouse in a apparatus so it can't move, and then you put your syringe in an apparatus that's fixed and has a motor, and it can deliver the needle exactly where you want it.
Yeah, you sort of come up with coordinates.
Yeah,
do
to make sure that you're in the right area of the brain.
Yeah.
Yes.
This paper made me very happy that I was in a biology department that is broad and has 3 neuroscientists.
Yeah, that's great.
Yeah. Well, they actually direct cancer chemotherapy like that with radiation as well. So there's a lot of things that work like that.
So in this paper, they actually, what they do is they add a gene to the rabies virus, which encodes the glycoprotein from a retrovirus, an avian retrovirus called envelope A. And then they will use that to infect to stereotactically infect the mice, for example. And then at the same time, they will provide a plasmid encoding the receptor for that glycoprotein because mice don't have it. So you can put the receptor and the virus where you want it, and that would limit the infection. So you get rid of the pathogenicity of the virus by removing the G glycoprotein. And they show in this paper that you can use it to trace the virus. They make fluorescently labeled rabies viruses, so you can use fluorescent microscopy. You can trace, and it does monosynaptic transmission, and it works really well. So this was like the first generation of this, and then subsequently people modified it. They learned how to make a G glycoprotein that was less cytopathic, better, more efficient at transsynaptic transmission. And they figured out how to make complementing cell lines where you would produce the modified G glycoprotein in a cell, and then you could use that to make your rabies virus stock. So you didn't have to add it in the mice. And then the mice, of course, will have the receptor already. And that modified glycoprotein, though, the problem with all of these so far is that they still are cytopathic. They still kill cells. And that's the problem that this current paper is dealing with, how to make a neuronal tracing vector that doesn't kill the neurons. I think the time you have with the old generation rabies viruses is what? 5 to 7 days, I think.
Where you get
that was
bit.
closer to 2 weeks.
Depends on where
5
you
to
get
17 days. 5 to
Yeah,
17.
that's what they say in the intro.
5 to 17 days. And then you'll see the vector they make in this paper, it's self-inactivating, so the mice don't die. Nothing dies in the mice. And that's the bottom line here. The neurons seem fine.
Dies.
The electricity is okay.
That's right.
Everything is okay.
Amazing.
We don't teach them anything, but so this vector is so cool. It's basically called SIR, self-inactivating rabies vector. And in fact, when I went to this talk at the IBC, she said, everyone's asking me for this, and we're just in the process of preparing it. You can buy vectors from this vector facility. Many places have these. And so this paper describes the production of this self-inactivating rabies virus. Now, part of this self-inactivation involves something we haven't talked about, which is targeting proteins for degradation by sending them to a complex in the cell called the proteasome. Kathy, can you explain that for us?
Sure. So the proteasome is a big complex of proteins, as Vincent said. Mainly, you could think of it as a cylinder, and then on each end of the cylinder, there's some additional proteins that are considered to be the gate. Inside the cylinder part of it are enzymes that can degrade proteins down to about 7 or 8 amino acid long little peptides that can then get degraded further. And the gate is mainly regulatory in determining what can get into this tubular proteolysis chamber. And the way that proteins get marked to be degraded by the proteasome is by attachment of a small protein called ubiquitin. And there's a whole, I don't want to say cottage industry. I don't want to offend people, but there's a whole lot of people who study all about ubiquitination and how the ubiquitin ligases attach these ubiquitins to proteins to mark them for degradation. But so that's a whole other area of regulation, that the protein has to be ubiquitinated, maybe polyubiquitinated is important, and then it can get degraded by this proteasome.
So it not only acts like a garbage disposal, it looks like one.
Yes, it does.
Now, Kathy, does something special have to happen to a particular protein before ubiquitin attaches?
Well, that's part of that so-called cottage industry that I was talking about. No, I mean, yes. So why
you can't do all of them, right? I mean,
why and how do proteins get tagged for degradation? So there are, it's a multi-protein reaction that determines the ligation and what should get ligated and so forth, right?
So the idea here is they want to say, can we stop the rabies infection by having the proteins shunted to the proteasome in some way? Okay. Well, that shut off replication. Will we have enough time to do something before it shuts off? All these things. So they basically put a signal on each of the rabies viral proteins by encoding it in the DNA, and it's called a PEST targeting domain. It's a sequence rich in proline, glutamic acid, serine, and threonine. And they basically attach the PEST domain to each protein, and between it is a sequence for a protease. It's a protease cleavage site from tobacco etch virus. And this just happens to be a protease that's good for these things. It's very specific. And so basically you could cut off the pest so it doesn't get degraded, or you could leave it on. So you can imagine this protease could be inducible in some way or deliverable by another vector, and then you could control how long the rabies virus was around, right? So they do a lot of work initially to figure out which rabies viral protein you can do this to, and it turns out that only one works. They put a pest next to each of the 6 viral proteins, and only putting it next to the N protein leads to conditional destabilization, as they say. They do this in cell culture. And so then if you add this virus, the proteasome degrades the viral protein, but if you have the protease present, it cleaves off the pest sequence and the virus keeps replicating. And they confirm that the proteasome is involved because they use an inhibitor of the proteasome. And they show that this inhibitor of the proteasome will prolong rabies virus replication, the one tagged on the N protein with pest. Go ahead.
I think the idea here is that rabies virus's RNA require some rabies proteins to replicate. And so we're getting rid of the RNA because we're getting rid of the proteins.
Right. So this N protein is involved in the replication of the genome and the production of mRNA. So you get rid of it, you don't have it, and then you stop replication. And so they basically show that idea works. You can target N to the proteasome, it'll inhibit replication. You can also remove N with the protease, and the virus will keep replicating. And if you have access to this paper, you can go to the methods where they talk about every step they took in the development of these vectors. It's amazing, beyond what they discuss in the actual. And they have a table with all the vectors they used. I think it goes on for 4 pages. It's unbelievable. I don't know if you guys noticed that, but it's really remarkable. So in the end, what they make is a rabies virus where the N protein has this pest sequence on it, and they've also stuck in the fluorescent protein called cherry, M-cherry, which will make a red color, by the way. Just kidding. It's a joke. It's cherry Dickson. Dickson's looking at me like... So they put these... First series of experiments are in cell cultures, and they want to know about the cytotoxicity of this virus. All right. So they did two more things. They added a destabilized cherry. So in other words, a sequence encoding for a cherry protein that will degrade with time. It's not around for a long time. And then they added a recombinase, a protein that recognizes a target and will cut it. It's called the Cre recombinase. And the system that's used is one we may have talked before. It's called Cre-LOX, where the Cre recombinase will recognize what are called LOX sites. And that was named simply because of LOX, the salmon thing. Okay.
See, I'm following all of this. And it sounds
so they
like
put
you're describing a meal.
They put this destabilized M-cherry into their rabies vector that we've just made and Cre recombinase. And they will inject this into mice that are transgenic for a yellow fluorescent protein, but it will not be produced unless the Cre recombinase cuts it and takes out a stop codon. Okay, basically. So without the Cre recombinase, no yellow. It's a switch to turn it on.
Yep.
So if the rabies is going to deliver the Cre recombinase, the yellow is going to come on.
And it turns it on permanently. For as long as the cell is alive, it will be expressing YFP.
It's permanent. Okay?
Cool.
So the idea is the rabies virus is going to go in, it's going to make cherry for a while, and then it's going to be shut down, remember, because it has the degradation sequence on it.
Auto-degrading, yes, sir.
So the cherry should go away, and it should be replaced by yellow.
The cherry will go away if the virus basically is loses activity.
Right.
If this is working correctly.
Mm-hmm. Okay.
The other color will come on if the virus was around long enough to express the Cre.
That's right. That's right. Yeah, so that's the question. We don't know if the timing is all going to be right, so they're doing this experiment. So I misspoke earlier. This is actually done in mice. This first experiment by injecting this rabies virus into the pyramidal layer of mice. At 3 days, post-infection, they only see red. All right, so that's the cherry
Cherry's
being made by the rabies virus.
Yep.
By 6 days, they see yellow. So the Cree is being expressed, the Cree recombinase, and they see yellow, and then no more red. And they monitor for cytotoxicity, so they look to see if the neuron's there for 6 months, no problem. The neurons are there for 6 months.
It's amazing.
The cherry is off, and there's no neuronal loss. And at the same time, they use the previous version of the vector, the deleted G protein rabies.
Yep.
And most of the neurons are dead within 2 weeks in the hippocampus, and none dead with this new virus. So this works.
Fantastic.
Isn't that incredible?
It is.
I wonder how long it took to get to that point.
You didn't mention the institution that they were part of.
Yeah, I did.
You did? Could you mention it again, please?
I'll have to scroll to the top. It's the MRC
the
Laboratory of Molecular Biology. Famous place, Cambridge, UK.
Right, because these are all Italian workers. I just thought that perhaps they were.
Yeah, no, they're at Cambridge.
All right.
Unlike other countries, the UK embraces immigration.
Exactly right.
Wait, then there's Brexit.
Exactly right.
Oh, yeah,
exactly
that's right.
right.
So that's
Exactly right.
Yeah, I'm sorry. Well, so now we have the proof of principle here, okay? And we have this vector that is inactivated. It can produce a modification of the mouse. It's cleaving this lock site by producing the Cree recombinase, and the neurons are not killed.
Right.
All right, so now they ask, can we control the degradation of the rabies virus, right? Remember, they had put this protease sequence in between the proteasome targeting sequence and the viral end protein. Can we modify that? So they use a second virus, an adeno-associated virus, to produce the protease in an inducible fashion. It can be induced with doxycycline. All right.
They also, a little bit later, talk about this AAV expressing TVA, and they never define TVA. Do you know what that is?
I looked that up for you.
That's the receptor for the avian leukosis virus glycoprotein that's encoded in these earlier versions of the rabies vector. So remember from the first paper, the way they did that was to put the envelope glycoprotein from this avian retrovirus into rabies, and then they also put a plasmid encoding TVA, which is the receptor for that glycoprotein.
Okay, so if I could just make a little aside, when we were talking about the first paper about readability, I wanted to point out that now that we have papers as PDFs, we can search PDFs for abbreviations. In the olden days, you could not do that easily. Some papers actually had the authors list all the abbreviations on the title page of the article. But they would still miss some. And then having a PDF only works when the abbreviation is spelled out somewhere in the paper, which it wasn't for TVA, and it also wasn't for another one that I pasted in the show notes a little bit later.
CHR2, channel rhodopsin.
Yes. Yeah.
Yeah, and also one virus I couldn't find in the paper. Another adenovirus wasn't described, I agree. But you know that you mentioned that, I always search the paper for abbreviations because I never remember what they are.
Yeah.
As I'm reading them, and I go, all right, I got to go back.
Yeah.
And this time I made a list in the show notes simply because I don't use them. And I'll forget them, right?
Good idea.
So just to tell the younger generation that it's bad, but it's not as bad as it used to be for us.
So TVA actually stands for Tennessee Valley Authority, but that's
exactly
another way to make the association.
All right. So back to this experiment. They take this rabies virus, inject it into mice, and then they feed the mice doxycycline for 2 days at 2 different time points, either 1 week or 2 weeks after infection.
Mm-hmm.
All right. Mm-hmm. So while at 1 week, giving them doxycycline increases the amount of red neurons because they're prolonging the replication of the virus because they're preventing the degradation of the end protein. So it works. You can actually feed mice doxycycline. You don't have to inject it. You could just feed it to them. It will get there and it will prevent the degradation of the rabies virus. But if you feed them doxy at 2 weeks, it's too late. Virus is already inactivated and you cannot increase. So there's a window where you can prevent this. If your experiment needed it for some reason, you can manipulate the degradation of the vector. So that's very cool. You can fine tune it. Right?
I really liked that they showed that the virus, was really gone here.
Yep.
They couldn't turn it back on.
Yes.
Right.
And they have lovely photos. The fluorescent photos of the neurons are beautiful.
Yes.
And the drawings are really nice too. The experiment we've described is actually here in a picture, and of course it's a lot easier to see in the picture, but on a podcast we can't show you.
Yeah, you're right. I hadn't really thought about that, but the figures are quite readable.
So the next question is, will this virus cross the synapse? Because that's what they're making it to do, right? The previous generation rabies vectors do cross synapses. All right, it's important to show that this does. So they do an experiment where they ask, will this virus cross synapses? And they compare it to the G protein deleted rabies. So the way they do this, they inject the nucleus accumbens as part of the brain. Okay? The nucleus accumbens, I'm not going to tell you what that is now because it will take me too much time. It's a part of the brain with an adeno-associated virus that produces TVA, which is the receptor for the retrovirus glycoprotein. And it also produces the rabies glycoprotein that I mentioned that's been optimized for efficient transsynaptic spreading. Okay. And that is their control.
Well, that just sets up that area as a target.
Right. They're not making any virus yet. They're just making proteins.
Right. That sets up that area as a target for any virus that's injected that has NV on it.
Right. And
the
then,
pseudotype with NV.
Then they infect with either their new virus, the self-inactivating rabies, or a G protein deleted envelope pseudotype rabies. That's the previous generation rabies. Okay. So their rabies virus, remember, uses this optimized glycoprotein to infect cells. It's provided by the adeno-associated vector, and the previous generation vector uses envelope A TVA interactions. Now,
and really, other than that, the only difference between this new virus and that one is this proteasome tag. Okay.
The proteasome tab and also the attachment, because the new virus is using the optimized rabies glycoprotein. The old one is using envelope A and TVA.
Well, now wait a minute, because neither of them have the rabies glycoprotein G.
Not in the genome, but they're provided on a plasmid.
Right. But the NV TVA thing is what targets either of these viruses to what I will call a proximal site, wherever they inject it. Okay. And since that, and that site will also contain this optimized G protein. And both of the viruses are going to use that, right?
No, I think only their new one uses the optimized. I think the old one, the Delta G rabies, uses envelope A and TVA to get into cells. Because if you look at this paragraph, then we retargeted the nucleus accumbens in the two hemispheres with either SIR, self-inactivating rabies, or Delta G rabies envelope A pseudotype viruses. The envelope A have to use TVA, which is provided by the adeno-associated virus that they already put in, and the SIR requires the optimized rabies glycoprotein.
Because am I looking at the right figure here? Figure
4.
Figure 4A.
Yeah. So does that say something different?
I'm looking at the cartoon.
Uh-huh.
And the cartoon has on either one hemisphere or the other, the injection of that same AAV TVA OG. Okay, so in both cases, you're getting expression of TVA and the optimized glycoprotein. And then according to this picture, on one side, you're injecting the old rabies virus that they're calling Delta G Rab GFP NVA. And on the other side is SIR CRE NVA.
Yeah,
so
right.
my interpretation of
they
that
both have
is
NVA, yeah.
is
but why do you?
They're using the TVA NVA system to establish the infection in the, what's that abbreviation stand for? The NAC?
Nucleus accumbens.
Nucleus accumbens. And then both viruses, according to my interpretation of this, use the optimized glycoprotein in the process of spread, okay, from there to wherever they're going. And the question is, do they travel in the same fashion? And do they travel as efficiently? And down the road, do the neurons survive?
I don't know why you'd need both, though.
My interpretation of this is that if you use the pseudotyped virus and the TVA, that establishes the infection in just one site.
Okay.
And from there, that way you know that any activity you're seeing at a distal site is because of actual travel from that original virus. I don't know if I'm articulating
it. No, it makes sense. Yeah, because if you put in an adeno-associated vector that produces TVA and the glycoprotein, that's only going to be made in the initial cells. And if you want the virus to spread, you need to enable that, because that virus is not going to be genetically, is not going to encode those proteins. So you have to support it.
So there's a sentence here that says the expression of TVA permits selective infection of the starting cells in the NAC, while the expression of OG allows its transsynaptic retrograde spread.
All right.
Bingo.
Correct.
So that distinguishes between the two. That way you know the spread is really spread.
So I'm sorry to have this long.
That's okay. This paper is a mind bender.
The bottom line here is that there's no difference in the spreading capabilities of the two vectors. Both can spread transsynaptically. And as we said, the delta G rabies drops out neurons. It kills neurons, whereas the SIR does not.
Mm-hmm.
That's very impressive data.
It's really beautiful. Yeah.
If you look downstream, if you look at the spread 1 week later, they look equivalent.
Yeah.
But if you look 3 weeks later, although you see the spread in both cases, there are fewer neurons in the case of the old vector because it's killing them.
Killing them, yeah. And that's in the nucleus accumbens. They repeat it in the hippocampus, basically, to show we can go somewhere else and have the same thing. And same idea, it can spread transsynaptically.
Okay, so now I've got a problem here. Okay, because let me understand this. The color here is results. Does the color result from expression of CRE in the virus, causing expression of tomato in the cells? Is that correct?
No,
the red I think does.
The red does. Okay, and the other one, the cyan, is just that virus expressing that protein. So here's my fundamental problem with this that maybe you can help me out with. What that means is, okay, so what is actually moving from cell to cell? It's the genome of the virus?
No, virus particles.
Virus particles. Okay? So I guess the fundamental thing for me is, in order to make virus particles, you have to have some transcriptional activity, right?
Mm-hmm.
This is what I would call a Princess Bride moment. There's this scene in The Princess Bride with Billy Crystal, where the dread pirate Roberts has been tortured to apparent death. They're trying to bring him back to life, and there's this big discussion about the difference between all dead and mostly dead.
Yes.
Okay, so I'm thinking the only way I can explain this is that this virus is not all dead. It's mostly dead.
Well, it can replicate enough, right? It can
replicate enough.
Yeah.
Right.
And if you put doxycycline in, you can extend the replication.
Right. Okay. So you get just enough so that you can express enough to make some particles and to make some CRE downstream and et cetera, but not enough to kill the neurons. So have I got that right?
Apparently, yeah.
Okay. That was my big issue with this whole thing. Because if it was all dead, then none of this would happen. It would you'd get a little bit of expression in the primary cell, and then you'd get no spread or anything. You got to have a little bit of expression to spread.
No, I mean, apparently that initial replication is not enough to kill the neuron. You need longer term, right?
Right. That was a big clarification for me. All right.
Now, one of the things they do is they say, okay, these experiments so far, we're using mice that have the integrated yellow fluorescent protein flanked by the Cre target sites. That not everybody will have that for their particular mice. So could we put Cre on? Could we put something on a virus and deliver it that would allow you to use this in any mouse line and not have to use a transgenic mouse line?
Right.
Yeah, I really liked that part. I thought it was written a little confusingly, but the idea of switching to the flip recombinase was a nice idea.
And so they use a different recombinase, and they make a adeno-associated virus that make this recombinase. Is that right? So
yes. That makes flip.
Makes flip. And they have another, and they also have another vector that encodes a nuclear tag GFP that has to be cleaved by flip in order to be produced, correct?
Correct.
And this works basically. When they put their rabies virus into the brain of mice, they can see the fluorescence in the host being made by this flip recombinase. So you don't need a transgenic mouse, in other words.
Exactly.
You can use vectors to deliver everything that you need in order to mark these animals. I'm trying to think of, is this permanent, this fluorescent protein production?
I believe so, because if they're using the Cre to modify the DNA of the cell, or they're using the flip recombinase. So it should be permanent in the neuron.
Because the adeno-associated vector probably also stays around forever also, right?
Yeah, that'll stay around for a long time.
So that might be part of it. All right, anyway, you can use your own mouse lines. So here's how the experiment works. They inject adeno-associated virus with TVA and the rabies glycoprotein, and then they inject adeno with this flip recombinase GFP in a known presynaptic area. And then eventually they see the color produced, which means that the rabies virus has reached that presynaptic area and spread. And this is all in non-transgenic mice.
Right.
Did I get that right?
Yeah.
Mm-hmm.
They also do an experiment showing that this virus can be used to study non-transsynaptic movement. In other words, within a single neuron. And they compare it with a virus that is called recombinant adeno-associated virus 2-retro. And of course, they don't explain what that is, and that requires you to go to the paper. So it turns out that adenovirus AAV2 will infect neurons, but in this paper, in another paper, they selected for an altered version of AAV2, which spreads really efficiently within single neurons. And that was done by mutagenizing the virus and putting it into neurons and selecting the ones that move the quickest and taking those and seeing what the changes were and going back and doing it over and over again. So this in vitro evolution type of experiment. Mm-hmm. So this AAV2 retro is used for studying intraneuronal movement, and they say basically their virus is as good as movement. It's more efficient in labeling subcortical structures, but it is a little bit less efficient in labeling cortical targets. But it does function as well as AAV2 as a non-transsynaptic tracer. Now, they do some studies to show that there are no long-term effects of this virus on neuronal physiology. They do a number of electrophysiological patch clamp studies, showing that there is no effect of the vector on functional connectivity and synaptic function in these mice. All right.
And they go 5 months out.
5 months out. Yes, right. 1 week and 5 months after injection. And then they do a very cool experiment where they say, can we use this virus for long-term circuit manipulations? And to do that, they want to make sure that the virus doesn't interfere with neural activity in vivo. And so what they do is they take anesthetized, immobilized mice, and they expose them to, they give them some visual stimulation, a moving grating field. And then
this is as close as it comes to taking them to the opera.
So the mice are looking at a screen with lines moving up and down, and at the same time, they are being imaged by 2-photon microscopy to look at the calcium dynamics in a specific part of their brain, which will change when they see the lines moving on the screen.
Right.
Okay, and they show that the infection with the rabies virus doesn't affect that. And so and in fact, you can watch the fluorescence change, the fluorescence induced by the infection, right? As these mice are watching right, Brianne, is that what you were going to say?
That's what I was going to say. And they also actually show that they can get sort of slightly different traces from different neurons, as if they can start to dissect out which neuron is responding to which pattern.
Right. So, when you insert the virus and you turn on fluorescent protein production, that is within synaptically connected neurons. So you label the first and the second neuron, and you can see what changes that, right? Do new synapses form and are disrupted all the time, right?
During learning, it surely does.
So,
absolutely.
So if you have fluorescence in a cell and it spreads to another cell, it could be by the establishment of a new synapse, right? And that could be, so you could look at mice in this way, or you could do anything to mice, behavioral changes, anything you want. And you could use this to say what's going on in the connections.
So one of the interesting connections might be for the use of this technology to show the effects of pollutants on learning behavior.
Sure.
For instance, lead is known to interfere with learning behavior. How does it do that? I'm told that it does it by actually gluing the synapses in place so they're difficult to disengage and reform.
Right.
So that this would be absolutely stunning to see that in living action.
Now, here's the cool part of this. How did they measure the calcium changes? They have an adeno-associated virus that delivers a protein that, when calcium binds it, it fluoresces. You can buy this.
Amazing.
I went and looked it up because I didn't know what this virus was. They didn't explain it. And you can buy them. They're called, what are they called? Biosensor viruses. Isn't that incredible? The fluorescence is activated by calcium or any other things, and that's how they can monitor this in these mice. So I think that's the long-term beauty of this, because the virus can modify the neurons and it doesn't kill them. You can, and I hate to use this word, but I'm just quoting the authors, you can functionally interrogate neural circuits.
You can label cells and then see what happens to the labeled cells.
Yeah, when you do different things like learning or running or swimming, whatever you can think of. And this is so cool that I said, I wrote in the notes, if I were starting science today, I would work on this. Because there's so many neurons, even if everyone on Earth worked on them, there'd still be plenty to do.
Well, now you can...
You know what you could do, too? You could infect them with viruses.
I was just going to say polio.
That are pathological and see what they're doing.
Vincent, as a non-virologist, we just had a common thought together here. Your virus, polio, you could find out exactly what the mechanism for pathology is now by looking at the way neural transmission occurs.
I couldn't, because the technologies, I have no idea, and I'd have to learn them, right?
This is like a set of spanners that you can order to do the repair of a certain, or just to investigate the function of. This is amazing. Just
so the other thing
amazing.
They say here, which is cool, this virus makes it possible to knock out genes of interest in a circuit-specific manner via genomic recombination of a conditional cassette. I'm not sure how you would do that. Does anyone understand that?
Oh.
So I think the idea would be that you would deliver your virus to maybe one neuron and hope that you're getting transsynaptic spread. And so you would just be labeling that sort of.
But how would you knock out genes in those?
You would use the flip
recombinase?
recombinase. You'd use the flip recombinase.
But then you'd have to have a flip target in the gene that you wanted to disrupt, right?
Right. And that's why they had to do, in their flip recombinase system, they used one virus that brought in the recombinase and one virus that brought in the substrate.
Got it.
I wanted to back up because I got a little lost in the weeds when you were talking about doing the flip recombinase versus the Cree recombinase. Because the way I understood it is that the reason they wanted to do the flip recombinase is so that they would be able to use this with all kinds of pre-existing mouse lines that have Cree driving expression in different tissues. And so I pasted in the Cree driver network page for neuroscience research. But essentially, because they built the thing originally with Cree, that would preclude using all these mouse Cree driver lines that target selected neuronal populations. And so they had to change it to the other recombinase, the flip, so that then they could go back and use this Cree driver system, mouse lines that are already out there.
Yeah, I would, frankly, I'm a little confused by that as well. I mean, the justification for those experiments, they say the use of Cree recombinase in our self-inactivating rabies limits its application in combination with available mouse Cree lines.
Right. So the available mouse Cree lines are these more than 100 Cree driver lines that have been created that target selected neuronal populations in the brain.
So then they say this is a limiting factor when trying to initiate the transsynaptic spreading from genetically defined neuronal populations. To overcome this shortcoming, we used flip recombinase.
Right.
So that in that case, they put the flip recombinase in the rabies virus, right? No, that was in the AAV.
AAV.
They put the flip in AAV, yeah.
No, wait.
I got very confused reading parts of this as well.
We generated an SIR, that's the rabies virus, encoding
with the flip.
A flip recombinase.
Right.
And then they have an adeno-associated flip GFP, which only makes GFP when the flip recombinase is delivered.
Is around.
So that's sort of a reporter, I think.
Yeah.
Yes.
But I don't understand how, that's a proof of principle, but how would you actually use it? Because if you want to disrupt a gene, it has to have...
Oh, we're talking about two different things.
Okay.
Yeah, well,
we're not talking about disrupting the gene, and I'm going back to what they originally put as their proof of principle, confirming that they would be able to use the SIR flip in conjunction with available Cree mouse lines, expanding the range of the use of the SIR.
So does flip work with Cree?
It does not. It works with the thing that they label as FRT.
So is there an FRT set of mouse lines as well? Is that what we're thinking here?
No, I don't think so. No.
Because they're Cree lines where the target has been placed around various genes, right?
Mm-hmm.
And that's
no. Do you mean that the Cree recombinase is there, or do you mean that LOX-P is around the genes?
So they say available mouse Cree lines. So that these are mice that make Cree recombinase. Is that correct? Or they have LOX sites at specific places?
That was my area of confusion, or one of my areas of confusion.
I think they make Cree in different tissues based on what promoter they're by.
Right. I think so too.
In the end, they say these results confirm the efficiency of a Cree and flip-dependent combinatorial approach based on the use of SIR flip in conjunction with available Cree mouse lines, of expanding the range of use of the SIR. Mm-hmm. So it sounds like flip worked with Cree or
No. The flip works for their rabies. In other words, whatever they made to their rabies virus in the first place with Cree, they've gone back and redone it.
Got it.
But now with flip
All
instead.
Right. It's just a reporter.
Right.
It's a way of getting there. Instead of using the yellow in the Cree, in the LOX position in the mouse, they use flip, which can be delivered.
Yes, exactly. Bingo.
Okay. Well, folks, this is how science works.
Yeah. Well, as I was reading this paper, I was thinking, it's as hard to do by myself. I'm looking forward to the TWiV discussion, even though it might be confusing, because it needs a journal club. You need several heads to get together on this and figure out what's going on.
Chalk talk.
Right. So I'm going to try and start the summary of this, or for the higher level view of why we found this interesting and why it's important. And something that didn't really come up, it's right off the bat in their abstract. It's becoming clear that network dysfunction is at the core of a number of psychiatric and neurodegenerative disorders. And so understanding these networks, and Vincent quoted to you all kinds of statistics about how many things each neuron is connected to, that is the next big complicated project. And this virus allows you to do that, because you can, a virus like this has been around for about 10 years, but the problem with it originally was that it eventually kills the cells. And now they have figured out a way to make this virus so that it doesn't kill the cells. And they do all kinds of things to show that what they've done to this virus doesn't change any of the neural function that they want to study.
Right. So you can essentially inject this virus somewhere and watch it move. And that, in effect, maps these networks.
So you can map the networks, but you can also label specific monosynaptically connected neurons, and then do things to the mice and see how the connections change.
Mm-hmm.
Exactly.
Right. So they've basically built tools that other neuroscientists are going to be able to use to map the brain.
Exactly.
Yeah, that's totally why I think it's cool. I mean, I'm amazed that you can engineer viruses to do this. Right?
Multiple
when
viruses.
we first made infectious DNA copies of we were happy to just change an amino acid here and there. But you could take genes out. You could put other ones in that have nothing to do with the virus. You can add other viruses at the same time. It's just amazing.
Tools.
And so the basic virus here is a rabies virus. Some of the experimentation is done delivering an engineered adeno-associated virus as well. The whole TEV protease thing comes from a plant virus. And the pseudotyped rabies virus receptor combination, that's from a murine leukemia virus.
Mm-hmm.
Okay? So there's little bits and pieces of viruses everywhere that are put together to make all this happen.
Anybody who doubts the importance of basic science research should read this paper.
Absolutely. This was made possible by basic research on these viruses.
That's right.
For sure.
And why are you working on a tobacco mosaic virus, for God's sakes? Smoking is bad for you.
And, I have to say that when people did these initial basic virological studies, they weren't thinking about making a tracer.
Of course not.
Right. Did we mention that Cree is also viral from a bacteriophage?
That's right.
Good point. Yeah.
And what about Flip? Where's Flip from?
Flip is from Saccharomyces cerevisiae.
Huh.
Oh, why should we work on yeast, right?
That's right.
Doesn't cause
That's right.
Saccharomyces. It just makes beer and wine.
Yep.
All right. Well, that is such a cool paper. I love it. Sorry if we confused you. Unfortunately, it's behind a paywall, I think. But I just think it's amazing. And I really look forward to, I'm not sure I can understand the papers that will come out of it. But
I can just hear these people in their planning stages for these experiments saying, no, we could use, but what if we used, what are we trying to do? What's the purpose of this?
The
We have to do
thing,
this first.
There are not that many authors on this paper.
Yeah, that's the point, though. That
and there's a lot of work here.
They must have had all of this like in a dream. They woke up one day. I know, I got it. I know what to do. Come on, let's go do it. Yeah. And the next thing they did it.
The other thing is this. We might not normally do this because it's making a vector, and we don't typically look at that, but the possibilities here are so amazing. All right.
You know what? This is not behind a paywall.
It's not cool.
No.
Because you're at home and
I'm at home. I can turn on. I can turn on and off my
VPN.
My VPN, and I've got it off now so that I can Skype with you guys, and I can get this article up.
Look at that.
Isn't that wonderful?
I tell you.
Let me do a few emails. Rich, can you take that first one?
Sure. Let me scan down here. Okay. RP writes, this is Alan Dove and Rich Condit's disingenuous response / topic hijack. Dear TWiVians, the weather here is 64 degrees Fahrenheit and partly cloudy, cool enough to open wide the windows in the library and enjoy the sounds of the birds. I'm a great fan of the shows. In fact, I subscribe to TWiV, TWIP, and TWIM, so I keep up my end. For your numbers. Forgive the temerity, but just in case you and your listeners are not aware, certain podcast platforms often unsubscribe listeners who are late to listen. 2 episodes piled up, unplayed, trigger, and unsubscribe. So I always make certain to keep an eye out lest my schedule trigger an unwarranted drop from feed, which will negatively impact the ad revenue.
Thank you very much. We appreciate it.
Wow.
I didn't know that.
Yeah,
I didn't know that either.
Mine doesn't. I mean, I have.
Mine does not either, yeah.
But I believe it. Yeah.
Even as a young child, I was raised to be unbearably conscious of revenue to production costs being directly proportional to stress-free days at night. That said, these specific podcasts are special to me, and I thank you for all your hard work and dedication and the hours of edutainment you provide. I write today because I have grown to respect the panel and was disappointed to hear what I must consider a classic misdirect as to the issue of organic versus non-organic food. To pretend the question is one of nutritional equivalency is to ignore the fact that one is laden with pesticide and the other is not. To proclaim that it is more efficient to slowly poison the population who live near farms and poison those who eat to consistently sicken actual farm workers exposed to poisonous pesticide is such a dark ends justifies the means dismissal. I prefer to let you all follow that thought through to its conclusion. It is a shame that our organic labeling laws are riddled with workarounds, but that does not make the concept of organic simply a marketing ploy. As for the GMO labels, I am as frustrated as any educated consumer who sees non-GMO verified labels on foods, especially when most labeled have no corresponding GMO on the market. I am even more frustrated by those around me who are afraid of science or believe they can simultaneously care for their health whilst ignoring the science and scientific methodology. It is even more personal for me than most. I am a person who was recently legitimately diagnosed with allergies to a couple known chemicals by an actual panel of real medical doctors. As it turns out, the chemicals are commonly used as biocides, preservatives, and pesticides. This was no easy task. Quackery abounds. More time, tests, and money, ad hoc research, and loss of life quality than I care to mention. Listening to TWiV helped me through some painful sleepless nights, but I digress. When it is posited that labeling for cholesterol or carbohydrates is more important than labeling for pesticide, or labeling for peanuts is more important than labeling for pesticide, preservatives, or biocides, is to ignore the science. I am not alone. In fact, one of my allergies is more common to laboratory professionals and others than many would like to learn. Unfortunately, for our uneducated letter writer, he is also wrong on certain crucial points. Organic is not kinder. Organic can mean no pesticide grain fed to animals which endure torture. There is no animal welfare rating attached to the label organic. Organic food can be harvested by slave labor. Organic is kinder to the farmers and the farm workers only in that there are no pesticides poisoning them. And last I checked, it was kinder to keep them in the field working rather than in the emergency room or the insert illness of choice here ward of the hospital and out of the grave. If pesticide purveyors were forced to pay health care bills and environmental cleanup costs and to handle the logistics of administering the care and the cleanup, they would not be efficient. Perhaps someone on the panel would like to do the math on that. Thank you again.
Dickson, is that right? Is there a high cost to pesticide use as the writers?
There's a tremendously high cost to pay for it regarding its...
No, we're talking about healthcare, environmental cleanup costs.
Well, yeah, to try to get rid of it is very difficult indeed. And that's the reason why a lot of, I guess, mythology is associated with organic food growing, because you can be honestly diligent about where you're going to plant your crops, and you've done a good analysis of the soil to make sure that none of these things are there. And then the next thing you know, your neighbor who doesn't agree with you takes a little monoplane and goes right over their crops with a pesticide, and that blows right over into your crops. Your organic farm is right next to theirs, and the next thing you know, even unwittingly, that pesticide is now in yours as well. So, and the analysis isn't often done, so it does end up on the consumer's plate, and that's the biggest problem with trying to label all these things without doing the analysis. You know, to say you're an organic farmer doesn't mean that your food is safe.
Well, I'm sorry Alan isn't here to comment on this, because I'm sure he could talk at length about both the organic and the GMO issues. I am significantly less educated on this, and my only gripe about either of these issues, organic and GMO, is the sort of blanket rejection of genetically modified organisms.
Right.
Okay.
Right.
I think that is there's a sort of a big paranoia fear thing that is being cooked up when the science says that genetically modified organisms or genetically modified plants in particular we're talking about bear no real risk. Okay. Now that, as we've already talked about, that doesn't mean that Monsanto are the good guys, right? There are all sorts of problems with the whole industry of genetic modification of organisms that need to be addressed. But the organisms themselves, so far as I know, so far are no problem. And in fact, they help with this whole pesticide issue. Okay. So that's all good. So as I said, my only problem with the whole thing is the blanket rejection of genetic modification of organisms.
Yeah.
I don't know what we said last time, but I trust that RP was turned off by something we said about organic versus non-organic.
I guess yes,
but
I think Alan was talking about it and said that there's no nutritional difference.
Correct.
Between organic and non-organic.
That is
food.
what he said.
Okay.
That is
and what RP is saying is that organic is about more than that. It's about the pesticides and stuff. And that's, as far as I know, that's a legitimate point.
Sure.
All right, let's do one more. Kathy, can you take the next one?
Sure. Alexander writes, Dear TWiVers, I'm a microbiologist researching Brucella in Berlin, Germany. The weather today switches between rainy and sunny, back and forth, 13 degrees Celsius, 55.4 degrees Fahrenheit. Humidity 89%. Winter, autumn is coming. I count myself among the group of more recent TWiV listeners, so I'm catching up on some past episodes to get settled into the podcast. In TWiV 455, Pork and Genes, Kathy Spindler mentioned something concerning severe viral respiratory infections in children with IFIH1 mutations, which struck my philosophical side. She said children with this mutation may have something genetically wrong, and I'm pretty sure all of us understand what she is really meaning by that. Nonetheless, did I ask myself if we as scientists should judge? I feel like calling this mutation wrong is not appropriate. It's probably a regular part of the human gene pool. No one really knows, A, if it might once have been the wild type, and B, if people might have some other benefits from it. I am aware that this example is extendable to recognized illnesses such as hemophilia or red-green colorblindness, which might complicate the matter a lot. Do not misunderstand me that the IFIH1 mutation leads to children going to ICUs is awful, but classifying mutations as good, bad, wrong, right, or else tends to add a moral point of view, which I am not sure if it suits a scientific evaluation, as it is not neutral. In the end, people and their genomes are different. The world would be a very sad place if this were otherwise. I hope I could get my message across. What are your thoughts about that? Are we able to judge or classify? Should or shouldn't we? Must we? Keep up the good work. Best regards, Alexander. So, I don't quite remember the context in which I said this, but he's quoting me as saying children with this mutation must have something genetically wrong. And I guess I was just that's kind of a self-referential statement. Children with mutation have something wrong genetically with respect to that mutation, which seems to have a deleterious effect with respect to viral respiratory infections. And so I totally agree that to say something is a particular genetic makeup is wrong or right or good or bad is really not appropriate. In fact, when I do my science communication things, I've had people describe what is wild type. And that becomes very difficult to do, as we all know. That
I knew a few in college.
Well, that's a different kind of wild type. But so I totally agree that, and what I said in that context was probably not well thought out.
I think you were trying to be what our first paper told us to do today, to be
simple and
simple. I interpreted that you were speaking to a broader audience.
Yeah, right.
But I agree with you and with the writer that we absolutely don't want to judge or classify or any of that based on mutation. Yeah, for the reasons that he said. Yeah,
I agree pretty strongly as well. I actually often have trouble with the idea of showing students that just because nucleic acids have changed, that there is a mutation doesn't mean that something terrible is going to happen. That's actually a really hard thing for them to understand. A lot of them have the idea of, you have a mutation and then you die. I think that trying to separate mutation and bad is important.
Yep. Environmental context is everything, isn't it?
It's like saying people who are left-handed were sinister, right?
No, I think about sickle cell anemia and sickle cell trait as a good example of a balanced polymorphism that actually benefits people living in high transmission areas for malaria. You move those people out of that environment into non-malaria areas, and then it's disastrous. And then that's, it has a horrible effect on their outcome. And so it depends on the context of the environment that you talk about. This mutation may have had tremendous selective advantage at one point for some other reason, not related to whether you're more susceptible or not to respiratory infections. So
just remember, mutation is a change in nucleic acid.
Absolutely.
That's all it is.
Absolutely.
And obviously you're comparing it to something.
So the environment is the one that selects this mutation or not.
Yeah, they're right. We know they arise randomly.
They're just going to be there.
You and I are full of mutations,
exactly
right?
Right. You bet.
No, nothing implied. We're just trying to find out why some diseases are more severe in some people than others.
Exactly right.
I agree. Go ahead.
There is nothing, either good or bad, but thinking makes it so.
Who said that?
Yeah, who did say that?
Bard, Hamlet.
Oh, really?
Act 2, Scene 2.
Nice.
I love it. Well, John Steinbeck paraphrased that. There's nothing, there's no good and no bad. There's just things people do.
Yeah.
Is that a paraphrase? Oh, maybe
good
not.
Enough.
All right,
it's all a matter of perspective.
Let's do some picks. Dickson, do you have a pick?
I do, actually. I changed my pick from what I originally picked because it was pretty self-serving, actually. I've submitted some photographs.
So I didn't see the original.
Good, because we've erased that and put in something else. So the National Geographic every year runs a contest for various categories, and in this case, they're now running a contest for the Nature Photographer of the Year. And, this is a good way to get opinions on your own photography, basically. So I've submitted a number of photographs to it, and they had some likes and dislikes, I guess. But then they have a big file, all of the submissions, and you can go to that, all of the submissions, and scroll down, and some of them are absolutely stunning. It's very humbling to be part of this scene, to be honest with you. And I ran across this photographer, Will Aides, who has some of the most remarkable weather photographs I've ever seen. And so that's what this is. These are frightening to look at.
I'll say.
You look at these things and you say, "Oh my God, I'm going to die any minute now because it's going to come down and eat me." And he stands there with his camera and tries to adjust the light, and he's looking at composition. Meanwhile, the bolts of lightning are coming out of the sky, and the tornadoes are forming, and hail is falling, and it's remarkable stuff. And just this is the real world. This is not made up.
from Australia, too.
This is not a computer-generated image. This is the way the world really is. So that's a good follow-up on what we were just talking about. These are the mutations, and these are the results, and some of them are going to be favorable, and some of them are not. But in this case, this is nature altering the environment on a grand scale, and I'm just attracted like hell to it.
These are amazing.
Beautiful.
Yeah, my favorite, even though it's not really the most violent, is this Montana storm.
Yes.
It's just these fields,
Exactly.
just these vast fields with a little farmhouse and a couple of trees, and then this incredible
beautiful.
storm.
It's quite beautiful.
Yeah, it's really pretty.
Yeah.
There are others in this series that are amazing.
Yeah,
also.
if you scroll through this, and then, of course, if you go to the website, National Geographic, and then click on weather, you'll get all of the people who entered that arena, and you'll see some just mind-blowing photography.
There's some here that are worthy of you. There's this young boy with a goat, which is, I could see you taking that.
Yes. Well, thank you. That's very complimentary. Thank you so much. I didn't. But when you put yourself up for judging like this, either you have to have a very strong ego, or you say, I'm got a ghost of a chance anyway, but I just like to know what people think. So that's why I'm in it.
Brianne, what do you have for us?
I have a book that I am about halfway through right now. It's called Pale Rider, The Spanish Flu of 1918 and How It Changed the World.
Ah.
It's a new book that just came out about the 1918 flu. And I've been really enjoying it because it talks about sort of the history of the flu and talks about 1918 flu around the world, which I haven't heard as much of sort of what was going on internationally in, India and China with the flu. It's really a fascinating book.
Have you read the other? Have you read the other one? What was it called? I'm looking on my shelf.
I have The Great Influenza.
Yeah. Is this better than that?
I have.
I'm not a fan of that one.
So far, I've been pretty happy with it.
Yeah. I didn't like the part where the author of that other book said, influenza virus infects cells, changes its genome to DNA, and then it integrates into the nucleus. How can you make such a mistake?
No, that's not okay.
You could just Google and you would find the right answers. It's ridiculous.
Didn't have the right fact checkers.
Oh. And then, I wrote a blog post pointing out all these errors, and he got mad.
At who? Himself or you?
Me.
Oh, he got mad at you.
He didn't actually respond on my blog. He responded somewhere else.
That's terrible.
And he justified all the errors, basically. Amazing.
Wow.
Oh, really?
So I hope this is better.
So far, I have not found any errors like that.
You're going to have more bad press, Vincent.
Rich, what do you have for us?
I got a science article that Ed Niles sent to me because he found it at least amusing. This is entitled Supergenes Drive Evolution. And this grows out of a lot of genomic sequencing, and it describes a hereditary phenomenon that has to do with sequences, areas of the genome where an inversion has taken place. And I have to go back to my just ancient genetics, to especially fly genetics, to think about this.
Exactly.
But when that happens, what it means is that it reduces or eliminates the possibility that the genes in that inverted piece can undergo recombination during chromosome pairing and meiosis, and so they get inherited essentially unscrambled or unchanged en bloc, and it allows a sort of a specialized kind of evolution to go on in those genes where they're kind of on their own game rather than something else, and rather than playing along with all the other genes. And apparently you get some, because of this sort of specialized kind of evolution, you get some interesting phenotypes that arise. And the one that's featured in this article, they mentioned some others, is in these birds called ruffs. And ordinarily these birds have a typical pecking order where the biggest bully gets all of the females, right? The biggest, prettiest bully gets all the females. But there's a block of this flipped inverted DNA that can control the appearance in these birds in a couple of different ways. One is has a white feathered collar that must be attractive in some fashion, but otherwise he's not really a bully or an ordinary bird. But he must be of some advantage to the bullies, because if he kind of hangs around with them, he can get his share of the action.
He looks like something from Revolutionary Times or something, like the wigged guys.
Wow.
And there's another one, and this is the best for me, that makes the male look like a female. So he can sort of mix in the crowd and get his mates surreptitiously.
Look at that. That's amazing.
No,
Amazing.
I just saw this in my own science.
So there's a couple of other examples, and honeybees and some other things.
Very cool.
Lots of
Really
interesting looking birds in their own right.
Could be a TWiVo story.
Could be a TWiVo.
Yes, as a matter of fact, add up to L's. That's exactly right.
Yep.
Good TWiVo story.
Kathy, what do you have?
Well, we talked a lot about the eclipse before it happened, and there have been some pretty cool things that have come out since. These were both as astronomy pictures of the day. One is a still image that's today's astronomy picture of the day. It's a solar graph. So it was an image that was made with, a drink can pinhole camera and light sensitive paper. And so I thought that was really creative. And then a little further back, there was a very cool video made of totality. And I like not only the video part that you see, but the soundtrack of people just getting so excited about what they're seeing. I think it's the best eclipse video that I've seen. So
it's very cool. I really liked it.
It's beautiful.
Yeah.
The solar flares are amazing.
Amazing flares.
Yeah.
Especially when the camera goes around to the top.
Yeah.
Oh, they're so big.
Yes.
Yeah.
They are.
That's great. Hey, that was an amazing thing. 2024, Buffalo, New York.
Yep.
Be there.
Austin, Texas.
I'm excited.
Cleveland, Ohio.
Yeah, that's right.
Austin.
Austin, Texas. This is right. This is right.
Who's left? Me? Everyone's done.
Yeah,
right?
you're left.
My pick is an article in a blog called Idle Words, whose tagline is "Brevity is for the weak," which I just love. So this is by someone who writes very, very long articles. And this is an article about the nonsense that happened on Amazon lately. British Channel 4 ran a news segment with the headline, "Potentially Deadly Bomb Ingredients Are Frequently Bought Together on Amazon." And this is just nonsense, all right? And in the article, it goes through why it's nonsense, and why the news organization should have done a little bit of research to realize why this is nonsense, their conclusion for this. And he says, it'll never get fixed. It's hard to fix any of this stuff. So he explains the science. And the real thing I want to get to here is the final analysis. And he says, this is a problem with news reporting, and it's not new to this. It's happened very many times before. Let me just quote some of this. "The real story in this mess is not the threat that algorithms pose to Amazon shoppers, but the threat that algorithms pose to journalism. By forcing reporters to optimize every story for clicks, not giving them time to check or contextualize their reporting, and requiring them to race to publish follow-on articles on every topic, the clickbait economics of online media encourage carelessness and drama. This is particularly true for technical topics outside the reporter's area of expertise, and reporters have no choice but to chase clicks. Because Google and Facebook have a duopoly on online advertising, the only measure of success in publishing is whether a story goes viral on social media. Corrections, if they happen at all, are quietly inserted through ninja edits after the fact. There's no penalty for making mistakes, but there is enormous pressure to frame stories in whatever way maximizes page views." And it says at the end here, where is that wonderful quote? Where is it? Here. "As the old adage has it, a lie can get halfway around the world while the truth is pulling its boots on." And, this is so true, and it applies for every area that's written on. But, I'm thinking of it in terms of science. This is why so much bad science gets promulgated, because people don't spend enough time checking stories. publish something wrong, and then they don't correct it. And they're publishing to try and get clicks. So this is a real problem. Somebody has to do something about this because you cannot compete with Google and Facebook unless you make something really, really clickable. And I think it's kind of sad.
That quote has got to be Mark Twain.
You think?
I'm going to look it up.
Yeah, you just type it in.
I would call this the Fox News syndrome.
Well, unfortunately, it's not just Fox that does it.
Yeah,
they all do it.
I know.
And that Amazon for bomb story was one of them. It's just nonsense.
Yeah,
total nonsense.
How do you feel, by the way, when you go to Amazon and order something and you've scanned the categories first, of course, to get to the right one, and then you buy something? And the next time you go online, no matter what it is, you see a little pop-up ad for something that you looked at.
Whatever you've looked at, you see on other sites.
I feel like somebody's looking over my shoulder all the time. I really do.
That is the income system of online. That's how it works.
That is not good.
And it's terrible. I go to some sites and I want to read something, and this big thing comes up in front of me.
Exactly.
And on some computers, I can't even get rid of it. It won't even move.
Real.
It's unbelievable. And then I say goodbye.
Or you go to that item and then you can't get back to where you were on the same website that you were on before. You have to go right back. Yeah, sure.
It's a real problem.
It's terrible. It's very annoying.
So we don't put any ads on any of our sites because of that. I think it's annoying.
Yeah,
and
it is annoying. You're right.
So
yep.
Anyway, the writing is very good at that lovely site.
I really enjoyed that. It's really very good.
He's got some other cool articles too. He took a trip to Antarctica, which is a great description. And the image at the top of the page is some kind of a fish, Dickson.
I'm not on that page. I'm looking at something else that you'd be interested in talking about in a minute.
Well, I'm about to wrap it up. What do you want?
Aren't you going to describe the picture in front of us?
Oh, that picture of me?
Yes.
Oh, yeah, right. Thank you.
From my other pick.
So, Brianne, a couple of weeks ago, you picked Shapeways, the viruses there, right? So I bought a couple, as I said I would. I limited myself to 3.
Yes. But one of them is strategically placed exactly where it should be. I love that. That's a great...
I didn't even think of that.
That's fantastic, Vincent. That was amazing.
Oh, you actually looked.
Yeah, of course. I'm looking at it
So I have
now.
3, and I put them on the table and photographed them with me behind, and the rhinovirus happens to be in front of my nose.
Exactly right.
That's
perfect.
Did you see what Paul Bianash called it?
Yeah.
What did he say?
Oh, that was so funny. I don't have it up right now. Do you?
I might be able to pull it up. But I did find a site that's called quoteinvestigator.com, which investigates whether Mark Twain said that quote.
And he did not.
Too long of an article for me to summarize here. So,
right.
He did not. I found the same thing. He did not. It can be attributed to Jonathan Swift, perhaps, but there's a whole bunch of related phrases that evolved over time.
Right.
Anyway,
okay, so Bianash's big green clown nose virus, BGCNV.
Nice. I had polio, rhino, and Zika.
With
What?
The wall of polio in the background.
Polio, nice reflection on the table.
He's got a pair of earrings coming off.
It took a long time to do that.
To get it just
but
right,
unfortunately, the viruses are not in focus. It's focused on me. I couldn't get it to focus on the virus, even though I tapped it, and I took 20 versions, and then none of them were in focus. So
you need to have a camera with a bigger depth of field.
Exactly.
A real camera?
A real camera.
I think the phone should be able to do it if I could focus it on the virus particles. I wouldn't mind if I were out of focus,
It's not so bad.
Anyway, that site is fabulous. I emailed them and asked them if they could make an enterovirus 68, the other virus we work on. And they said, yeah, just send us a VRML2 file. And so I'm working on that. But
sure.
The problem is that I made one and it's like 10 feet in diameter. So I have to figure that would cost thousands of dollars. So I have to figure out how to shrink it. But there's so many beautiful viruses there, and Simon on Twitter said, how come there's no turnip crinkle virus? I said, yeah, there is. Here it is.
That's right.
They got lots of them. They're beautiful. Thank you.
They really are beautiful. I'm so impressed.
Influenza virus should look nice too, with all the projections coming off it.
I would like to make, the problem is we don't have a structure of the particle that might be useful. So you have to have, it's based on the structure, right? Cryo-EM or X-ray.
Yeah.
We don't have one for the flu particle.
Right.
I'd like to get a printer and just crank these out, myself, instead of having to buy them. And some of the bigger ones, Brianne, are like hundreds of dollars.
They are, yes. I have looked into doing a little bit of 3D printing. Our math and computer science department has a 3D printer.
Mm-hmm.
And I think it was much more complex than I expected.
Yeah, it is complicated. That's why these online sites are thriving, where you can send them.
And these are colorized too, according to their positions.
Yeah, you can color the image file on your like I use MacPyMole, and you color the chains as you want them. And but here, of course, they've picked the colors for you, but they happen to be correct. So, the VPs of polio and the E-glycoprotein of Zika are correct, but the rhino green these are not the canonical colors. So, no. But it's green snot, I figured, right?
Are all the flaviviruses yellow?
They should be, right?
That's true.
Well, this is a flavi, but it doesn't
got
affect
some
the liver,
yellow,
right?
some
Yeah.
yellow.
All right, that's TWiV 461. You can find it at Apple Podcasts, microbe.tv/TWiV. Send us your questions and comments, TWiV@microbe.tv. And if you want to support us financially, microbe.tv/contribute. Dickson Despommier can be found all over the web. Just amazing. For today, we will say thelivingliver.org. That's pretty cool. And parasiteswithoutborders.com. Yep. Thank you, Dickson.
You're welcome.
I'm sorry the paper was complicated.
No, I enjoyed it. Because the conclusions.
The problem is that I can see you and I can tell when you're not happy.
No,
I was very happy. I have no idea.
I was very happy. I was struggling with the details, you're right. But in the end, the conclusions were quite remarkable.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Brianne Barker is at Drew University. She's on Twitter as BioProf Barker. Thank you, Brianne.
Thanks for letting me be a part of this. I had a great time.
Yeah, pleasure. Whenever you're available, we'd love to have you.
Great.
And Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiV. I'd like to thank the sponsor of this episode, the Defense Threat Reduction Agency, and I'd like to thank Ronald Jenkins. He did the introductory music. You can find his work at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.